this stage, as I understand it, we're going to have a discussion between the speakers. So I'd like to encourage you all just to think about what you might have learned, sort of a higher level insights into mechanism and diversity. And who wants to kick off?
I have a question for Sandra.
Please.
Sandra, you mentioned that most of the domesticated plants cluster in a specific region of this global spectrum of plant diversity. Is that a coincidence, or is that these plants have some traits that facilitate their domestication or their capacity to produce food?
My impression is that it's not a coincidence that they were being first selected because they originally in the wild types originally had some desirable traits, and they were then further selected towards desirable traits. So, for example, the fact that the plants are relatively small, the plants, more importantly, rather than being small, they grow fast. Most of the plants we grow, most of the plants on that corner of the spectrum grow very, very fast, and they have very palatable and very acquisitive leaves, but the leaves don't last very much. And they do have nitrogen in somewhere, either nitrogen in the leaves, which made them very nutritious to our animals, and they have all these alkaloids, which we use for various recreational or medicinal or whatever use. And they are easy to grow during early agriculture, right? You just plant the seeds and you have plenty of germination. So those were the traits first filtered from the wild. And some of them are wild, some of them are weeds. All the agricultural weeds are in that corner too. And then, of course, we perfected the traits we wanted. We made the seeds bigger. So that's a corner in which our sculpting of phenotypes has been strong. Actually, the only corner in which it has been strong.
This is very interesting because there is a lot of discussion that we have limited our food production to a very small percentage of plant diversity, and that we should expand to use many more plant species for food production. So do you think that's feasible, or this tell us that biologically we have already selected the best candidates?
No, I think that if you remember, I showed 2 hotspots. There is one hotspot which are all herbaceous plants, and then there is another hotspot which are mid-sized trees with big seeds. That hotspot has already been tapped because those are trees that don't grow too slowly and they produce big fruit. The body of the plant itself is not very practical to us. It's not very palatable. It takes a long time to grow, but then they have these really nutritious fruits. And most of the tropical fruit trees are in that particular spot. Now, the other areas of the spectrum, the problem they have is that they don't grow fast. So it's probably not very practical to us. They are good for standing incredibly hard environments. So if we are interested in taking a page from nature on how you get a plant which is incredibly adapted to a tough environment, then we should be exploring the southern and southeastern corners of the spectrum.
I was going to slightly change the discussion, because what was interesting to me, Luis, is that you approach the desiccation with the RNA-seq and the network analysis. And a lot of people are doing that now for flowering, because obviously if they want to do it in different plant species, rather than go through all the full genetics, they tend to do RNA-seq. And actually the RNA, the networks that come out, you can see the same regulators that we've discovered through the forward genetics, but I'm not sure you would have necessarily picked out the major nodes until you've done the natural variation analysis. And it's the natural variation, it's the evolution's tweaked the various important nodes. That's how we discovered FLC and major regulators of FLC rather than. So I wondered, have people done natural variation of desiccation tolerance and then mapped that onto your networks?
We are currently working with resurrection plants, doing a similar analysis comparing species that are naturally desiccation tolerant versus closely related species that are intolerant, and also with grasses, to compare their responses to naturally desiccation tolerant and intolerant grasses, and building the networks to see how they change during the evolution of plants, because that obviously is going to lead us to the central regulators.
Well, I think the intra-specific analysis may be faster, where you can actually do the cross and the genetics and the QTLs, as opposed to, just a lot of network analysis.
Yes, we are doing also the crosses. And it's not that easy to cross a Legionella, which is our best model to do this. But we are also trying to generate mutants that become now intolerant. So, but the thing is that the response of all plants is very similar. So the effector genes is the same family of genes that are involved. But yes, indeed, we still need to go backwards to find the central regulators, which actually might be these developmental regulators like LEG1. Yeah. Because LEG1 controls the two processes, both the transition from embryo to vegetative growth, which it suppresses, and the activation of desiccation tolerance. So what we think is that we are just below LEG1 in the activation of desiccation tolerance.
Great. Can I just, before we have more discussion, I just want to say to the audience, pretty soon we'll be switching to audience questions. And so far we've had 2, but we really love more. So I really encourage audience members to type their questions into the Q&A bar. Sandra.
Yeah, a question to Luis. It's a very at the macro level. You show this phylogenetic tree, so you obviously look at the bigger picture. Is desiccation tolerance cluster in any particular plant groups today? In terms of phylogeneity or in terms of growth forms?
No. There are dicotyledonous plants that are desiccation tolerant. There are monocotyledonous plants. There are many, it's distributed among many different plant species. So it's not that it's clustered and it generated many desiccation tolerant plants. That's why we say it has re-evolved independently 13 times, because they are in different branches of plant development.
So just on that, so I think Herrera-Estrella, I think, is the classic resurrection plant. I mean, have you looked at, there's a lot of expression data, I'm sure, available from Dorothea Bartels and people like that. So can you see the same kind of profile showing up during desiccation of Herrera-Estrella that you see during the time course you looked at in Arabidopsis seeds? And I think you can transform Herrera-Estrella. Could you go in and mutate the orthologs of the genes you've identified and see if that leaf tolerance of desiccation goes away, or if there's been maybe even duplication of genes so that there's some for seed desiccation and some for whole plant desiccation tolerance?
Yes, we have analyzed the Crateroctisma and the Selaginella expression data, and the networks are similar, but they are not identical. So there are some common elements, and some others are different transcription factors that belong to the same family, but change from one species to another. They are not direct orthologs. And in some cases, we get the incorporation of completely different transcription factors as nodes. So the rewiring, although seems to have a general trend, they are not identical in all cases. So that's something we are working with, and we have also developed the transformation of Selaginella. So we want to do knockout of these genes in Selaginella.
Great. I've got a question to Sandra, actually. So in terms of biodiversity and human impact, something that we've done over the last 150 years is to elevate CO2 levels. So you can imagine that the competitive advantage of C4 plants with a more efficient photosynthesis when it's 280 ppm is going to be a lot less than at 420 ppm or whatever we are now. So I guess the first question would be, if you classify plants by C3 and C4 plants within those principal component analyses you displayed, do you see a pattern? Is any evidence for that pattern changing as CO2 levels go up?
I'll answer in two bits. First, there's no much clustering of C4 on the spectrum. It is slightly more common in the, say, the western part of the spectrum, simply because C4 is very common in grasses and other groups. It's not homogeneously distributed among families. And so it is there, but it is not associated. So having said that, C4, for example, if you look in grasses, you have C4 grasses, annual acquisitive, C4 grasses, extremely conservative, C4 grasses, very tall, very small. So it's kind of orthogonal, I would say, almost to this global spectrum.
Okay, thank you very much. I think time's pressed on, and we've overrun slightly on our intended time course. There's a few interesting questions from the Q&A. So Richie Perrick asks, I presume this is for Caroline, where are these signals detected? So is a detection of temperature equally in the leaves, in the shoot meristems, or in the roots? What do you know about where the detection happens?
The detection is in all over the plant, basically. That was the sort of picture I was trying to convey at the end, that all the cells are independently detecting and remembering those signals. And then it's combined into this sort of integrated effect through this mobile signal FT. So it's not, and this is very different from what people were expecting. There was an expectation it would be in the meristems or very specific parts of the plant, but certainly it's all of the, you know, the major growing parts, which is really the bulk of a small plant, is all detecting the temperature.
Thanks. So there's a question from Rob Martinson. I think I'll put it to Sandra first. So what do you think about perennialism, and where do perennials lie on this principal component analysis map that you showed us? And you just tell us, well, I guess the second part of the question is to Caroline, on what happens, how do flowering signals work in perennials? First, Sandra.
Sorry.
Perennials, do perennials and annuals map very differently in your...
Oh yes, sorry. Yes, definitely, space? Definitely. All the annuals are on the west. Yes, it's on the west, and it could be anywhere in the west. It could be northwest or southwest, but all of them are there. These are all the herbaceous and small plants, whereas there's zero annuals on the east of the spectrum.
Right.
Yeah.
So what about
my input onto perennials? So George Coupland's lab has been looking specifically at the basis of perennials in an Arabidopsis relative, and has come down to FLC regulation again. And it turns out, I think the simple answer would be, it's the SNPs within the gene itself, non-coding SNPs, that prevent the switch to the epigenetic silent state in that long-term memory. So there's a kind of slightly longer than metastable, but not long-term. And so the gene is reactivated always in the spring. And so some parts of the plant, which are old enough, basically when FLC is shut off in the winter, the positive signals actually start being expressed. those are then epigenetically on. So even when FLC comes back on, there's still that positive signal. But in other parts of the plant, because of an age-related pathway, those positive signals can't come on. So there's a beautiful combination between non-coding SNPs and FLC and an age requirement for vernalization to happen. And that can then explain the perenniality.
Can I just say something? Isn't the case that many annual plants have relatives that are perennial? For instance, sorghum has perennial relatives,
Yes.
maize has perennial relatives.
Yes, I think annualism has arisen from perenniality many times. And certainly in our data set, the annuals will have arisen from perennials.
No, but I mean in the map, if they separate completely, in the West, there must be many perennials.
Oh, many. In the West, there are many perennial. There are acquisitive, short-lived, small perennials, yes. So the perennials can be everywhere, but the annuals are only in the West. And I found the same as Caroline. Many annuals are rare members of mostly perennial families, with a couple of exceptions.
Okay, so that's the direction of evolution.
Yeah.
So I've got a question here for Luis. So could you compare desiccation tolerance to salinity tolerance, and could the approach you've taken to investigating desiccation tolerance also be applied to investigating salinity tolerance?
The response in the general response is similar in terms of the type of effector genes that are involved in salt tolerance. And some of the transcription factors that have been reported for salt tolerance are also part of the families or the same we find in this network. But I don't know if the network analysis has been completed to really know how similar the networks are. But in general, when you increase drought tolerance and desiccation tolerance, you get higher salinity tolerance. So the mechanism must be common in many components.
Another question for you, Luis. What about cacti? Do they actually have desiccation tolerance, or do their adaptation more to avoid exposure to desiccation?
Yeah, the cacti and many other desert plants, they have mechanisms of avoidance of desiccation. They store water, they have a different CO2 assimilation pathway. So they function in a different way to preserve and accumulate water to withstand dryness. It's not that they are desiccation tolerant.
Well, another question here from Jason Lynn. I think it seems to be a question about quantitative efficacy of your engineering desiccation tolerance compared to, let's call it the extreme desiccation tolerance of a Cratera stigma. So how far are you away from, engineering the transcription factors of Arabidopsis such that you could dry them out and completely and then revive them the way you can with the resurrection plant?
Well, we are probably 6 months away of activating all of them and see how far we can go in desiccation tolerance. As I mentioned, activation of 5 gives a very strong drought tolerance, but it doesn't confer complete desiccation tolerance, because if you keep this plant dry for a long time, they die. So we'll see in the next probably 6 to 12 months, I should know how far we can go in quantitative terms of desiccation tolerance.
Okay, got a question here to Caroline. I hope I can do justice to it. I'll just read it out. Since cold exposure memory is occurring on a population scale, is the robustness or accuracy of the vernalization decision depends on the size, i.e. number of cells in the root structures? And thanks for a great talk.
Thank you, Nicole. I think, my schematics inevitably had to be rather simple. So it's not just those few cells that are informing, it's a much broader set of cells. So I think it's more the probability of that switch then determines the proportion of the plant that is silenced FLC or not. And that is changed in natural variation. So that, you know, the SNPs at FLC do change that. The sand clock model, the aperture would be different depending on whether it's adapted to Sweden or France or wherever. But it's, I think, the number of cells rather than the size or the size of the meristem or anything like that, because it's a very large proportion of the plant that is silenced.